In part 2 of our video covering osmosis, we're going to talk about the concept of tonicity. Tonicity is a noun, and the adjective form of this word is tonic, although you don't normally see this adjective used on its own, because tonicity is a way to describe the difference in solute concentration that exists between two different locations. For example, if we refer to the solute in our situations as Na+, we can see that in this situation, the concentration of Na+ outside the cell is roughly equal or very close to the concentration of Na+ inside of this cell. So when describing the difference in solute concentration, we are describing the tonicity, and we can use this difference between solute concentration in two different locations in order to predict which way water will move via osmosis. Now, if we have a situation like this, where the solute concentration inside the cell is roughly equal to, or in some cases exactly equal to, the solute concentration outside the cell, this is what we call an isotonic situation. So here we can see the adjective form tonic is not normally used on its own. It's accompanied by this prefix here. So again, tonic is an adjective that refers to the solute concentration, and iso is a prefix that means the same. For example, an isotope is the same element, so same number of protons with a different number of neutrons, and an isomer is an organic chemical that has the same chemical formula with a different structure. So isotonic simply means same solute concentration inside the cell and outside of the cell. Now, what this means is that if we look at the percent water of each of these solutions, if the solute concentration is roughly the same, that means that the percent of each solution that is composed of water is also going to be roughly the same. Now, does this mean that water is not going to move in or out of the cell at all? That's not exactly what it means either. What it simply means is that the rate of osmosis into the cell, so the rate at which water enters the cell, is roughly equal to, or again, sometimes exactly equal to, the rate at which water will osmose out of the cell, meaning that the volume of water inside the cell and outside of the cell remains relatively constant, because water overall is not going to more easily move in either direction. Now, if we analyze this situation here, we can clearly see that the concentration of our solute inside the cell is significantly greater than the solute concentration that exists outside of the cell. And if we convert this into water terminology, the less solute we have, the greater the percentage of water is going to be. So this means that our high percentage water situation is outside the cell, and our low percentage water situation is inside of the cell, which means overall we would expect that more water is going to move into the cell than water is going to move outside of the cell. Now, this situation is what we call a hypotonic situation, although we should probably explain how to use this particular word here. Any word that refers to tonicity, especially hypotonic and its opposite hypertonic, are what we call comparative adjectives. So if we want to use this word accurately, we say that the outside of the cell is hypotonic relative to the inside of the cell, meaning that the outside has a lower concentration of solute and a higher percentage of water relative to the inside of the cell. And again, we can't use this adjective without a subject or without an object, because we're directly meant to compare which one has the lower solute concentration. Now, this is particularly dangerous for cells because this means that we're going to have a relatively large volume of water that is going to be entering this cell in order to decrease this solute concentration inside, which means that the cell volume, the amount of liquid inside of the cell, is going to perpetually increase. Now, is it possible for a cell membrane to hold this new water in? Well, maybe, but not forever, because as the volume increases, that means that the water pressure, the force exerted by this water, is going to be pushing outward on the cell membrane. And in extreme situations where a cell absorbs too much water, you can actually get the cell membrane to burst in a process that we call lysis. Again, this Greek word that we've seen in hydrolysis, and the verb form of this is to lyse, which simply means to break. Now, the opposite of a hypotonic situation is a hypertonic situation, and this is arguably just as bad for the cell. So here we can see that the area of high solute concentration this time is outside of the cell relative to the inside, which has a comparatively lower solute concentration, which means that the inside now is our high percentage water situation, because if we have less solute, then water makes up a greater percentage of this solution than on the outside, and therefore the outside would be our low percentage water situation. So naturally, in this case, if we have more water on the inside of the cell compared to on the outside of the cell, the rate of osmosis into the cell would now be less than the rate of water osmosis outside, which means overall we're going to have a net movement of water leaving the cell to go outside in order to decrease this solute concentration here. Now here, we can see that this is an obvious problem, because any cell that is placed in a very high solute solution is going to start losing water in order to drop this solute concentration outside. And this is why drinking ocean water is such a bad idea, because drinking ocean water contains a larger concentration of salt than what we have in our cells. And this is why people that drink ocean water often feel even more thirsty, because their cells are losing water in order to decrease the salt concentration from ocean water. Now, if you were to place a living cell onto pure sodium chloride with no water in it, you can actually get the situation that is happening here, where cells lose almost all of the water that they have and eventually desiccate, which is the scientific word for get killed by drying themselves out. So based on these two situations here, understanding what a hypotonic solution is compared to the cell and a hypertonic solution compared to the cell, we can see that both of these situations are equally deadly to the cell, and thus it is very important why a cell needs to maintain an isotonic balance of water and solute inside and outside the cell. Now that we've finished learning about the concept of osmosis, you can see if you can apply it to this case study here, where we actually talk about the unusual condition known as water poison or water poisoning or water intoxication, and identify, using how osmosis works, how people would actually harm themselves if they actually drink too much water.